Welcome to XenoTech's webinar, Transporters of Emerging Importance in Drug Development Beyond the Guidance Documents. My name is Michael Milhall, and I am the Global Marketing Manager here at XenoTech. Today I'm joined by Dr. Brian Ogilvie, our Vice President of Consulting. Brian received his Ph.D. in toxicology from the University of Kansas Medical Center. He has been with XenoTech since 1997 and currently serves as a Vice President of Consulting. Brian is an author or co-author on over 50 scientific posters, peer-reviewed publications, or book chapters on the topics of drug metabolism, transport, and drug-drug interactions, and has represented the company as an invited speaker at various drug metabolism conferences. As Vice President of Scientific Consulting, Dr. Ogilvie is additionally involved in quality management and strategic planning for the company in scientifically relevant areas. It is my pleasure to present to you Dr. Brian Ogilvie.
Okay, thank you, Michael, for the introduction, and thank you for everyone. Good morning or good afternoon, whichever part of the world you're in. I'm going to start out with a regulatory view of in vitro transporter studies, and then move into a little bit of a refresher for some of you, and maybe possibly the first time you've seen this, and I'll try to briefly cover it, the extended clearance concept system and the prediction of rate determining steps in ADSI, and that is not a typo. I will get back to why that is in a little bit. And then evidence for emerging transporters, and I've focused on OCT1, OATP2B1, and OAT2. And then finally, for unusual routes of administration, I have a number of clients that have worked on nasal and ophthalmic drugs in particular, but then also other routes that come into play, which I won't necessarily cover in great detail. So the current regulatory view of transporter studies, I'm going to basically take these figures from the most recent International Transporter Consortium 3 paper, which came out last year in CPT. So the way I'm going this is with, for instance, the intestinal epithelia here. You can see that the requirements are in red for FDA and PMDA for all BCRP and PGP. For EMA, it's orange, and I'll get to that in just a minute. But for substrate potential, we know FDA and PMDA require that for PGP and BCRP for all orally administered drugs. For hepatic uptake, and if you go here, now you can see orange already required, OCT1 by EMA, and green, in this case from the ITC paper, means that OCT1 should be evaluated prospectively at this point. That we then have the OATPs, 1B1 and 1B3. OAT2B1 is in yellow because the ITC3 recommends a retrospective evaluation of that I'll cover in more detail. And of course, the typical biliary secretion, so PGP again, BCRP, and MATE1. Now, if you look also for hepatic uptake and even the renal secretion, the guidance documents will talk about hepatic metabolism or biliary secretion are greater than 25%, or if unknown. Same with renal. So if renal secretion is greater than 25% of total clearance, or unknown, then you would need to look at that for substrate potential. So for the renal transporters, that includes at this point OCT2, OAT3, and then the MATEs. ITC3 recommended MRP2 and MRP4 as retrospective. And then if you look at the brain capillary endothelial cells, now we've got, of course, PGP and BCRP as we have for several years. There are other transporters, and I'll talk about a few of those at the very end. So timing, I want to cover a little bit of how this works from the FDA's perspective. Because the FDA split their guidance document in 2017 into one in vitro and one clinical guidance, timing is not mentioned in the in vitro document to any great extent. So you have to work backwards from the FDA clinical guidance. And the question is posed, when are clinical drug-drug interaction results needed? And that is before administration to patients. So when I have this in quotes, it's because I'm quoting directly from the guidance documents. I try not to paraphrase guidance. And within the clinical guidance, it says collect enough DDI information to prevent patients from being unnecessarily excluded. So that would come into play when you're talking about getting into your phase 2 or later programs, or if you're developing a drug in the oncology space or something else where you have to enroll patients in phase 1. So they go on to say, FDA does, that the inadequate studies of DDIs can hinder the FDA's ability to determine benefits and risks of a drug and result in restrictive labeling, post-marketing requirements or commitments, and/or delayed approval. But one of the things that's most important when it comes to clinical development is preventing patients from being unnecessarily excluded. So that in particular can occur during oncology development in your phase 1 study or later other drugs in phase 2. Now, the choice of transporters investigated should really be driven by scientific evidence, and this is underscored in the ITC 3 recommendations based on current scientific evidence. So from hepatic OCT1, again, as I mentioned, they recommend prospective evaluation for both inhibition and substrate potential at this point. And when it comes to intestinal OATP2B1, retrospective evaluation in specific instances of DDIs or disposition otherwise unexplained by more common mechanisms. MRP2 and 4 and BCEP, they were previously recommended for retrospective mechanistic explanation of clinical observations. ITC 3 is putting forward that there's not enough evidence at this point for a general recommendation on this going forward. And then, as I put it, an honorable mention for several other transporters, including OATP1A2, 4C1, OAT2, which I have highlighted because I'll go into more detail on that later, and then ASBT, OST alpha and beta, NTCP, even MDR3. They state the evidence is lacking for specific drug development recommendations. So that means not that you shouldn't look at those, but that when you have reason to, it makes sense to go ahead and do it, follow the evidence. So I want to, as I mentioned, briefly cover the extended clearance classification system because I'm going to be talking about some of these drugs in terms of their ECCS class. So the goals of the ECCS include earlier prediction of rate determining step in human absorption, distribution, clearance, and elimination. So that's where that ADC acronym comes in from, not ADME. And this goes back to papers from Ayman El-Katan, Manthana Varma, 2015, and then more recently from Ayman and Varma again in 2018 in DMD. And one that's in JPET right now, which is from, again, a Pfizer group, including Dave Rodriguez, a senior author, and it covers essentially SLC phenotyping in hepatocytes. And so I'll cover a little bit of that here. So if you're not familiar with the ECCS, I want to briefly walk you through this. So if you look at the y-axis, if you will, we can divide drugs into high and low permeable. And this was developed with a cutoff based in a low transporter expressing MDCK cell line that was selected over time. And then within each one of those, you can look on the x-axis, acids and zwitterions, and then bases and neutrals. So class 1 would include high permeable drugs that are acids and zwitterions. And then within that, you have this molecular weight cutoff of 400 Daltons. And anything less than that would be considered class 1A. So metabolism is most likely to be the rate determining step. If you have a greater than 400 molecular weight, it could be hepatic uptake, so class 1B. And then moving over again to the high permeable class 2 drugs, these are bases and neutrals, metabolism is expected to be the most likely rate determining step in human ADC. Going on to the low permeable acids and zwitterions, so again, the molecular weight cutoffs come into play here. And for renal clearance for class 3A, these are molecular weight less than 400. For molecular weight greater than 400 Daltons, now we're talking class 3B. This could be hepatic uptake or renal clearance. And then for class 4, this is mainly renal clearance for bases and neutrals. And this was the initial framework from 2015 and drawing on much earlier work as well. Now that the work has become more sophisticated and more cases have been put into play, And I'm drawing this figure directly from the Rodriguez and B paper that is in JPET right now. I add a little bit more to it. So for high permeable drugs, not only for class 1A and class 1B, you have this transporter enzyme interplay. So even though these are high permeable drugs, with the class 1A, you can potentially have OAT2 playing a role. And for class 1B, you can have OATPs and NTCP involved. We knew that hepatic uptake was already in play. This is just making it a little bit more specific. Now for class 2, again, I said earlier that metabolism would be expected to be the rate determining step. But now we have many cases of possible hepatic uptake by OCT1. And when it comes to these low permeable drugs, so class basically 3A, where you expect renal clearance to be the predominant, now we have, again, OAT2 showing up. Of course, OAT1 and 3 are showing up as they have in the past. And class 3B, where you have hepatic uptake or renal, it could be OAT3, OATPs, and NTCP. Now for class 4, again, these are bases and neutrals. For the renal clearance, it's still OAT1 and 3, OCT2, and the mates, as you would expect, but also a possibility for hepatic uptake by OCT1. So I'll get really to OCT1 relatively early, and I want to go through the basics of OCT1. It's the most highly expressed OCT in human liver. And if you look at on the Human Protein Atlas, they have these nice diagrams like this. I actually did not show the protein on the right side. I'm only going to focus on RNA, knowing some of the limitations there, but there are also limitations on the protein side depending on the methods used. But you can see that the liver and gallbladder really light up for RNA expression. We know it's also expressed in kidney to some extent as well. So in the liver, OCT1 is a sinusoidal transporter. It's facilitative, and it is actually highly polymorphic. And that was a little bit of a surprise to me when I first learned about this in 2015 or 16. It transports weak bases. So of course, we've known for a long time OCT1, metformin, but then also phenodorol, tropicitron, ondansetron, even ranitidine, and O-desmethyl-tramadol. So this is a metabolite of tramadol, essentially as a prodrug, and the active metabolite here. And then for triptans, I'll cover sumatriptan in some more detail here. That's why I have it highlighted. And so, as I mentioned, I really started becoming interested in this in 2015, and especially in 2016 when I saw this paper come out, that OCT1 is a high-capacity transporter of sumatriptan, and polymorphisms that cause OCT1 deficiency have similar effects on sumatriptan PK as those observed in subjects with liver impairment. So that really piqued my interest, and I had to study this paper. In fact, I even presented a couple of reviews of this in various presentations. And I've found in the literature in this paper and also through the University of Washington Drug Interaction Database, their ePK gene module, that there are 5 common OCT1 polymorphisms that are present in up to about 9% of Caucasian populations. So these polymorphisms, OCT star 2 through 6, have strong or nearly complete decrease in activity. Poor transporters have increased plasma concentration of tropicitron with a Cmax increase of about almost 400%. Sumatriptan, you see an increase in exposure of about 200%, as I mentioned, similar to hepatic impairment. And then with O-desmethyl-tramadol, as I mentioned, you see an increase of 74% exposure, and that is from tramadol as the parent drug. And again, another prodrug situation, so give codeine and you see an increase of morphine exposure by about 70%. Phenodorol, this is a more recent paper that's come out, if I remember correctly, about a 90% increase in exposure. And there are some hints for others, so including imatinib and the anti-epileptic drug lamotrigine. So I want to focus on sumatriptan. That was the paper that really piqued my interest, and I think it's one of the more interesting case studies. It was, of course, the first triptan on the market, and it is actually still very highly prescribed. It has a low bioavailability, and that's largely due to the fact that it's metabolized by MAOA in the liver, MAOA is a very rapid enzyme. It's not usually a rate-limiting step in metabolism like P450s are. It has a negative log D, and so it's about 95% cationic in blood. Has a half-life of about 2.5 hours, and only 3% is excreted unchanged, about 60% in the urine and 40% in the feces. And as you can see, it tends to, as I mentioned, have that permanent positive charge on it at physiological pH. So the data the authors of this paper in 2016 presented showed that MPP plus does inhibit sumatriptan uptake in human hepatocytes. And these are pretty convincing data. So if you look at 0.1, 1, and 9 micromolar sumatriptan, you can see the uptake, and it's knocked down pretty significantly by MPP plus. Sumatriptan uptake was also examined in HEC293 cells by either OCT1 overexpressing or OCT3. And you can see pretty clearly on this graph that the clearance, basically the intrinsic clearance, if you will, for OCT1 is much higher than OCT3. And surprisingly, it's also about 6-fold that reported for metformin in similar test systems for OCT1. They also looked at the clinical effect of OCT1 poor transport on sumatriptan. So the effect of loss of OCT1 function on sumatriptan PK, you can see that if you have 0 active OCT1 alleles, you have a dramatically increased exposure. And that translates then to about a 50% reduction in total oral clearance. And the way I think of this is that OCT1 controls access to hepatic MAOA. If you look at the AUC graph, you can see that the number of active OCT1 alleles, if it's 0, you have a dramatically increased exposure. But even if you only have 1, so heterozygotes, and we see this pretty frequently with transporter polymorphisms, it's not like the typical case with CYP2D6 where there's a wide range of polymorphisms. This is almost an all or nothing case. So you either lack it, and one active allele will allow you to have normal transport. But this is, as I mentioned, similar to the hepatically impaired patients, and also you see a 50% reduction of extrarenal clearance of sumatriptan in the OCT1, OCT2 knockout mice. So it's concordant with those other in vivo results in other species. So OCT1 mediated transport of other tryptans was also examined by the same authors. And they looked at in OCT1 overexpressing cells versus the control, that elatriptan you really don't see much effect. Naratriptan you certainly do. Some effect with rizatriptan, of course sumatriptan as I mentioned, and zolmitriptan. And there is an inverse correlation then between uptake and the PAMPA permeability. So you can see the graph here. You have the permeability on the x-axis and then the OCT1 transport ratio. Here's sumatriptan, and then naratriptan is up here. Elatriptan, rizatriptan are down here on this part of the axis because they have a higher permeability, as you would expect for many transporter substrates. Now, the interesting thing about the polymorphisms is that it can be substrate dependent. So although I mentioned about 9% of various Caucasian populations are poor transporters for OCT1, only 2% of Europeans and white Americans are poor OCT1 transporters with respect to sumatriptan. So substrate dependent effects, different polymorphisms actually affect O-desmethyltramadol, morphine, and metformin, as well as sumatriptan differently. So if you look at the in vitro results that these authors presented with the different polymorphisms, you can see quite different. So like OCT1 star 2, for instance, there's essentially no effect of that polymorphism on sumatriptan uptake in that in vitro system. And there are some other minor alleles that were also examined. So for instance, the star 5, star 6, and so on. So potential drug-drug interactions, the main evidence so far with sumatriptan is obviously with MAO inhibitors, as you would expect from its metabolism. But there is also some evidence for increased efficacy of sumatriptan at lower starting doses with the OCT poor transporter. So the question arises, if you inhibit OCT1, would you have potential DDI? And honestly, that's not well described at this point. Elatriptan and rizatriptan would be less likely to have OCT1 mediated interactions because of their higher permeability, but I think, again, this is a possibility. Naratriptan, I'm interested in that one because it does have a lower permeability, and it would be interesting to look at the FDA's adverse events reporting system to see if there are results with naratriptan that have been reported. For instance, we know that metformin has interactions because the pharmacodynamics are relatively easy to measure. However, if you look at strong inhibitors of OCT1, you don't see much of a change in the exposure, systemic exposure of metformin. And this is for a variety of reasons, in part because the liver is the main organ of efficacy, so it's easy to measure the pharmacodynamics, and also because there is significant renal clearance through OCT1. And I'm going to move on to OATP2B1. There's a little bit less data on this, but I'll cover it as much as I can. It is, again, the most highly expressed OATP in human intestine. And you can see from the RNA expression that it shows up in a variety of organs. Liver and gallbladder and gastrointestinal tract certainly appear, but it's also in other organs such as lung and so on. Now, whether or not those mRNA expression relate directly to active protein and the proper localization, that's another question. So in the intestine, it's apical and sinusoidal in the hepatic uptake. It's polymorphic as well, and it transports several drugs. So I've got the statins, rosuvastatin or torvastatin, pravastatin, but then also other drugs like celiprolol, fexofenadine, montelukast, aliskiren. And then if you look at the impacts of polymorphisms and DDIs, there are several polymorphisms, but they're not particularly well characterized yet. There are, in fact, if you look at the University of Washington Drug Interaction Database, the EPK gene, I found variable and sometimes contradictory effects. So for instance, they show that with poor transporters, if you will, of OAT2B1, that you see an increase of rosuvastatin of about 112%. Celiprolol, you actually see a decrease in AUC, about 50%. And then a differential effect on S-fexofenadine versus the racemic fexofenadine, you see an increase or a decrease. And again, this may depend on the particular paper that was cited. Montelukast, you see a decrease in exposure of about 46%. And with drug-drug interactions with OAT2B1, they are complex. So if you look at ronacaleret, inhibits intestinal OATP2B1 and therefore decreases rosuvastatin AUC by about 50%. On the other hand, asuprinovir inhibits hepatic OATPs and therefore increases rosuvastatin AUC by about almost 200%. So depending on if you're inhibiting the intestinal versus hepatic OATP2B1, you can see a differential effect. And you'll notice that down here, asuprinovir, I mentioned OATPs, and that's because ITC3 and Mashek in particular, when he presents this, will note, and I recommend also that if a drug candidate is a strong OATP1B1, 1B3 inhibitor in a definitive study, that sponsors recommend or consider at least an OATP2B1 inhibition study. And this is mainly because of the promiscuity of OATP inhibitors. I have seen some clients that have very selective OATP1B1 and 1B3 inhibitors, but they're not typically found commercially available. So in other words, the point here being that if you see inhibition of both OATP1B1, 1B3, you really should consider looking at OATP2B1 to make sure you won't have a potentially unexpected interaction. Now, OAT2, I mentioned that earlier, and I'm wondering if this is sort of just the beginning of the story. This is an interesting story for me. It goes back to 2015, 2016. Andrea Wolf, who is still part of Sekisui XenoTech in collaboration with several Pfizer authors, including Dave Rodriguez, published this paper in 2017, and we looked at OAT2 and OAT7. And if you go looking for OAT7, you'll find even less about that. But this most recent paper from last year, and there are some more recent papers in OAT2, to me, this is a really groundbreaking paper. They're looking at basically the OAT2 mediated uptake contributing to the clearance of high permeability, low molecular weight acids and Zwitter ions. And they use 25 drugs, but I think there are other examples as well. And one working hypothesis would be that essentially all high permeability, low molecular weight acids and Zwitter ion drugs may be OAT2 transport substrates. So what about OAT2? It actually happens to be the most highly expressed OAT2 in the liver, OAT just in general, in the liver in its sinusoidal expression. You can also see that it's expressed in the kidney and urinary bladder, as we already knew. And there it's basolateral. It transports several high permeability, so low molecular weight acids and Zwitter ions, as I mentioned. So this would be permeability limited ECCS class 1A drugs. And data have come out more recently. There was a paper from, again, Pfizer authors, including Manthan and Varma in 2018 in JPET, and in some more recent papers mentioning S and R warfarin. There's a differential effect there. Also tolbutamide, and then other NSAIDs, so diclofenac, phenoprofen, ibuprofen, ketoprofen, even going on to some of the isoxicams, meloxicam and piroxicam, but even including some of the glitazones and tolcapone and gliclazide. So OAT2, I looked for polymorphisms. I can't really seem to find any. I haven't been looking on a daily basis, so it could be that there's a paper that's come out. But this is something that I think is going to become more and more frequently reported in the future. Drug-drug interactions. So there are many DDIs with the OAT2 substrates that are often attributed to CYP2C and UDT inhibition. However, when you look at PBPK models that take into account the enzyme transporter interplay, so CYP2C, OAT2 in particular, they provide better prediction. And I think this has intriguing implications. In fact, if you look back at tolbutamide and some of the other drugs that I mentioned, you tend to underpredict the in vivo clearance when you're looking only at microsomal clearance, where you're looking at CYP2C. So one possibility of a drug-drug interaction, there's some evidence that decreased theophylline clearance with erythromycin is due to OAT2 inhibition. And then there's also even some data that, for instance, there's an interaction between warfarin and indomethacin. Now we know that indomethacin will also inhibit CYP2Cs, but the IC50 of indomethacin toward OAT2 with creatinine as the substrate is as low as 2.1 micromolar. Then there's another sort of intriguing finding in the literature that interferon alpha 2B downregulates OAT2 and actually potentiates warfarin. So now going on to unusual routes of administration. And this comes from some of the studies that we've done in the past with a client that had a nasal spray, and they started out with two separate nasal sprays that they were going to then combine. And I can't talk about this because of the proprietary nature, at least in detail, but what they found clinically was that there actually was an interaction that was unexpected when these were both administered in the same nasal spray. And in that case, it appeared that possibly OCT1, 2, and 3, and maybe OCTNs and several others may be at play here. We never got a definitive answer from the study. And there's also complicating nasal administration, something called the nose-to-brain transport through the olfactory and trigeminal nerves. I'll show you a slide here in a minute that walks through that in a little bit more detail. And then going on to ophthalmics, I've had this question come up a number of times. You know, we're developing an ophthalmic indication. We're wondering if there are any sorts of transporters that we need to look at. And depending on the paper, there are a lot of transporters. So I'm just going to list them out here, and then I'll go into more detail in a few slides. So OATPs, several of them that we don't normally think of, and then OATs as well. And then some of the ones that we consider to be more hepatic, so ASBT NTCP, and then again OCTs, OCTNs, and the MATEs and the PEPs. So the nasal overview. So within the nasal epithelium, there are, as I mentioned, the OCTs, OCTNs, and others, but you have to consider the nose to brain transport. So for instance, if you look, there's the olfactory pathway and then the trigeminal pathway, as I mentioned. And when a drug can go into the nerve itself or go through the paracellular pathway, it can end up in lymphatic vessels, it can end up in the blood, it can then end up in the cerebral spinal fluid and then the brain interstitial fluid. Same with the trigeminal pathway and lymphatic and then on to the CSF. Olfactory mucosa itself, as I mentioned, has different transporters and then even respiratory, but I'm not going to cover that at this point. But you can have cases where in the nose to brain transport, the drug will end up crossing the leaky choroid plexus to end up in the cerebral spinal fluid. And then you have to look at a number of transporters there in the blood CSF barrier, which can include OATs, OATPs, OCTs, MRPs, and several others. And then the blood-brain barrier, of course, we're most familiar with the PGP and BCRP, but there are others. So OCTNs, again, pop up, OATP1A2, even ENT, and some others. Now, this paper goes into a nice review of nasal delivery, and that's where this figure comes from. It doesn't necessarily cover the transporters. You have to look at other references to find some of those, but it does mention it or refer to other papers that cover that. Now for the ophthalmic, again, I just wanted to give you a visual at how complicated this can be. So if you're looking at the ciliary body, there's this entire list of transporters that have been in various papers described as being expressed. This review came out in Drug Metabolism and Disposition in 2016. That's where I'm taking this from. And they list a number of papers. Mostly this is, the results are from mRNA expression. So again, taking with a grain of salt what that means in terms of actual expression and activity, but it gives you some idea. So within the iris, now we're looking at mainly MRPs and even BCRP. In the iris ciliary body, you have, again, a long list of MRPs and even PGP. So in other words, it's complicated. And if you go in more detail, the ciliary body and the iris, a deeper dive, you can look at more of these transporters, even including some of the ones I've mentioned before, OATP2B1, for instance, and some of the other OATPs that may or may not be commercially available. Within the iris, you have the different tight junctions and capillaries, OATP2B1, sorry, 2A1 and 2B1 both. However, the subcellular localization is unknown at this point. So to conclude, guidance is guidance, and it's there for a reason. It's sort of a minimal, and you may need to go beyond that in many cases. It depends on your particular program and what you know about the compound, and really it comes down to a risk-based assessment. How much risk are you willing to tolerate? And I do think the EMA probably had it right even as early as 2012. That guideline was finalized in 2001 with a routine evaluation of OCT1, which is now recommended in the ITC. So consider the ITC 3 recommendations and also the ECCS. Can you predict what can be involved? So class 2 and even 4 ECCS drugs may be OCT1 substrates, and that may be important in the hepatic uptake. And as I mentioned earlier, could it be that essentially all ECCS1A drugs are OAT2 substrates? I think this is something that we're going to have to start investigating further as a field. And OATP2B1 and then other transporters as needed. As I said before, it depends on what the evidence shows and what your program may have disconnects between the in vitro and in vivo as you go further in development. And then for special routes of administration, basically know the transporters in those tissues. Sometimes you really have to dig for this information. It may come down to even basic studies, even just looking at the tissue. If you know that this is something unusual, it may be that if you have unexplained results, unexplained interactions, as I mentioned, with 2 drugs being administered at the same time in a nasal spray, and you see unusual clinical results, you may have to really start digging into other reasons why you see those results. So thank you for your attention, and I'd be happy to take any questions at this time.
Thank you, Brian. And while we wait for questions, I wanted to mention that information about our services, papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Brian's presentation or any of Seksui XenoTech's contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website.
Okay, so we do have a few questions here. One question was commenting that OAT2 substrates are high permeability, and basically were the permeability studies also affected by the transporters? So in theory, when I say that they are considered high permeability, it's based on the ECCS papers. And what Pfizer did, and I think others have also as well, is used clonal MDCK cell lines that were selected over multiple generations for low endogenous transporter expression with a permeability cutoff of around 5 × 10⁻⁶ centimeters per second. So in theory, the effect of transporters in these permeabilities should be minimal. Okay, so then here's another one on the OAT2. So in our overexpression OAT2 cell lines, what ratio do you get with substrates? So with tritiated substrates, including cGMP, we have a ratio of about 30-fold. We use penciclovir about 35-fold, and then again, cyclovir about 30-fold. And I believe these are published in that 2017 Xenobiotic paper that I had listed. Looks like, okay, here's another question. Are potent clinical OAT2 inhibitors described? So at this point, not that we know of clinically, however, ketoprofen turns out to be a good in vitro inhibitor. And the recent paper from Pfizer that's in JPET, fast forward, I think it came out in mid-April. It talks about SLC phenotyping in human hepatocytes. So they used a range of concentrations of ketoprofen, showing that it is relatively selective for OAT2. Again, for clinical inhibitors of OAT2, there's no great evidence. There is a potential interaction, as I mentioned, between warfarin and indomethacin, but that could also be related to CYP2C9 inhibition. So that's really kind of up in the air yet, and I will be interested to hear if others in the audience or if there are new publications that show clinical interaction that can be proven through OAT2. I don't know if you have any over there on that side already.
Yep, so as a reminder, we will be sending out the presentation to approved registrants after the webinar. A question that we've gotten, with high permeability, would you expect the in vivo impact of being an OAT2 substrate to be low?
Surprisingly, no, and I say that because of the fact that drawing on multiple lines of evidence, but the fact I mentioned earlier, and there could be, that there is a confounding variable somewhere, but the evidence right now is pointing to the fact that when you look at human liver microsomal studies, which are, have for a long time been pretty predictive in determining in vivo clearance for many drugs, that with drugs like tobutamide and even warfarin and diclofenac, you tend to underpredict the in vivo clearance. However, when you include, and this again is coming from some of these recent papers that I referred to in passing, from Pfizer mainly, is that when you include the OAT2 uptake, then you can more fully explain and recover the actual clearance in your PBPK modeling by including that transporter enzyme interplay.
What is the typical MW range for the OAT2 substrates?
So the typical molecular weight range, because these are ECCS class 1A drugs, would be less than 400 molecular weight, Dalton's molecular weight, and that's just kind of a rule of thumb. That's based on empirically when the first ECCS papers were coming out, and then Pfizer, authors like Manthan Varma and Ayman El-Khattan and some others looked at a very large data set of around 600 drugs, and they found this relatively clear cutoff in molecular weight. And then some of the more recent data have borne that out with the OAT2 work that was published last year, and then some more papers more recently.
What are the intestinal versus hepatic OATP2B1 inhibitors used in clinical DDI studies?
So for OAT2B1, as far as clinical studies, I don't know that there can be a specific recommendation at this point. It was interesting. I think I had the slide earlier, and I don't necessarily need to show it right now, but just to remind myself, it was aciprinavir, and bear with me just a minute. So Runacalaret, and I'm sure I'm butchering that name, I believe that was a paper out of GSK, if I'm not mistaken, but I did draw that from the University of Washington database. That inhibited intestinal OAT2B1, so you see a decrease in rosuvastatin AUC, and with aciprinavir, that inhibits the hepatic OATPs. But again, I don't know that there is, or if there is, I've forgotten, honestly, what the specific inhibitors are for intestinal versus hepatic OATP2B1. And I would be hesitant, even based on just a couple of papers, to make a recommendation, especially since the ITC3 went through in great detail. And I don't recall a specific recommendation there, but I may just simply be not remembering right now.
All right, this wraps up today's session. Thank you to all of you for taking time out of your day to join us, and thank you to Brian for that great presentation. Have a great rest of your day.